So this presentation is going to be all about retinal processing. We've dealt with like retinal structure, laminar structure, how the whole thing's set up in the eye. Now the concept of how does that information get processed, at least in the eye? What are the initial indicators that get passed out of the eye? We'll get into all the details. We'll get into some things that kind of play a major role in how the retina starts to give the brain the right information to process. So one of the things that's key here is that only the ganglion cells are the things actually producing action potentials in the neurons. So there's actually no action potentials being formed in the bipolar cells, the horizontal cells, or in the photoreceptors themselves. Okay, they're just going to regulate action. They're going to regulate neurotransmitter release through depolarization or hyperpolarization. The ganglion cells are actually going to produce that action potential output of the retina that's going to travel through into the brain for further processing. So here, and you see that here too, we're going to produce created changes in memory potential for the horizontal bipolar cells, but so it's ganglion cells that are going to be our output. So just a quick reminder again, we've got our photoreceptors down here. Okay. When they get hit, they're going to release less neurotransmitter onto everything above it, okay? Which means the bipolar cells will get less of the neurotransmitter coming out of the photoreceptor. The horizontal cells will get less of whatever neurotransmitter is coming out of the photoreceptor, and that will cause an activation of some kind, but not an activation of an action potential, an activation of some kind of neurotransmitter release up here, this next synapse, which is going to be what causes a ganglion cell to change its activity levels. So a couple of things about retinal processing. First thing is receptive field. What is a receptive field? So we'll talk about this term a lot. A receptive field is just the area that responds to light in a given location. So somewhere in space, in the visual world around this animal, whatever it is, okay, these photoreceptors right here, okay, are going to receive input from a specific location in that world. So the receptive field for this bipolar cell down here are the photoreceptors that are going to send signals to it. The ones that are going to transmit their signal this way to that bipolar cell. They're the ones that are part of its receptive field. Now, what you can also see here, though, is too, we have input coming from other photoreceptors through this horizontal cell, okay, coming in this way, which is also going to interact with our bipolar cell. So what ends up happening is in the receptive field, in the photoreceptors that activate or change the activity of this bipolar cell, we are going to have two types of bipolar cells, one called an on-center and one called an off-center bipolar cell. What that means is a bipolar cell will be more responsive to light either in the center of its receptive field, okay, or in the surround of its receptive field. An on-center bipolar cell will respond to light in the center. An off-center bipolar cell will respond to light in the edges with greater activity levels. We get more to this in a second. But this idea is you have these antagonistic areas, okay, and that's going to be great for detecting things like edges, contrast, right? Okay, you can imagine, we'll get to that more later, but you can imagine as something moves across, like a light moves across this way. If we have a cell that responds to light in the center, and this is a darker object, okay, and it's moving across my retina, here, okay, it hasn't blocked this out yet, so this is still getting lots of light, still saying it's an on-center cell, it's still getting a lot of excitation, but now all of a sudden it gets blocked by this new object, it comes into there, and that's going to reduce the amount of excitation that happens onto this bipolar cell. And this is pretty consistent actually throughout the retina, and you'll see that in a little bit. So again, here we have that on-center bipolar cell. Light on, we get less glutamate response again, because remember, we're getting hyperpolarized when that happens. Now there is more depolarization by polar cell when the photoreceptor is hyperpolarized. Okay, so it's going to release more, but in terms of this synapse right here, we're getting less glutamate being released. We turn the light off, and more glutamate gets released because this interior photoreceptor here maintains its depolarized state. Whereas the light in the surround, okay, actually causes these other ones to stop releasing their neurotransmitter, and that ends up meaning that will end up resulting in the effect of the neurotransmitter from this cell being greater on that bipolar cell. So there's this whole idea of the surrounding cells are going to have an influence on the output of the bipolar cell because of these horizontal cells, down here causing the surrounding areas to have an influence on what that bipolar cell experiences. So these are ganglion cell receptive fields. So there's some tutorials here. If you want to go back and look at the slides later, you can do that. But kind of the idea of an off-center ganglion cell right here. So here you see you're in the patch of retina. You're in the receptive field for this particular ganglion cell. It's receiving input from like multiple bipolar cells coming onto it. Okay, and see here where the whole retina is lit up, you get a little bit of activity, right? You get like 3 action potentials in that region. Now, since it's an off-center ganglion cell, off-center on surround, okay, when we go into the middle of its receptive field, the dark spot, okay, when that spot becomes dark here in the middle of that receptive field, it hits the maximal peak thing here. Okay, it's an off-center ganglion cell, so it wants that center to be off to get its maximal activity, and that's what we see in terms of action potentials that record from the ganglion cell. Now, the other advantage too is we've also stimulated the on-surround too. This isn't just the off-center, off-surround. This is an off-center, on-surround. That on-surround's light, so that's going to contribute to that, which we can see in part C, where in the center we have our off, which is causing activity levels to go up, but we are also in the surround off as well, which is going to cause our activity levels to go down. So we end up with that combination there. Now you see, based on this activity versus this activity, the off center is a little more preferential versus the on surround. So the center seems to have a bigger influence on the number of action potentials than the surrounding does. And where this play a role, again, is where we get to edges, where you start to see things move across. You see here we have that same off-center ganglion cell. We get light in the middle, which is going to activate it, or which is going to inhibit it, but we have light in the outside, which is going to activate it. So we get a few action potentials here and there, right? Now, so we get a dark edge coming in. So now we're getting inhibition from the outside and inhibition from the center. Right? So both of these are slowing activity, and all of a sudden we get no action potentials whatsoever. Okay, so now in this case we had that on surround, but we're inhibiting that on surround because we put a dark in there. We took light away. Now all of a sudden you see the whole center gets blocked out. That's going to raise activity here, and this is going to lower, but you can see that preference is for that center. The preference is for raising activity based on that surround being there. And we're getting a little bit of excitation from this spot down here too. But now the black moves and covers the whole thing. We get raise an activity level here, but here, everywhere else in the surround, okay, we're getting a lowering, and that combination results in action potentials at a lower rate. Again, seeing here and here, more action potentials based on when just that center is lit, when just that center is off, showing a preference for the center of that receptive field. Okay, so a couple other things here for the retinal output. for ganglion cells, we're going to have two major types of ganglion cells. We're going to have an M-type ganglion cell and a P-type ganglion cell. There are non-MP type cells as well, too, but just so we can kind of see this in terms of what the output's going to be. So here we have our M-type ganglion cell called magnocellular ganglion cells. And see, there's an on-center cell. So when the light turns on, we get a really drastic increase in action potentials, okay? Really high firing frequency initially, but you can see that that starts to trail off, and as it goes along, it adapts to that thing, that light being on. So it stops sending action potentials. However, in the P-type ganglion cells, these are the parvocellular, we get an increase again when we hit the receptive field center, okay, with our increase in light. And you see there's a slight adaptation, but it's slow. And it actually keeps firing action potentials even though while that light is staying on. So these are two of the major types of ganglion cells. And to kind of the next slide, it's going to talk about some of the benefits to all of those things. So in those magnocellular large M-type cells, they're going to have larger receptive fields. Okay, they're going to be large cells with large receptive fields. I mean, they're going to respond to light from all sorts of different locations. They're also going to conduct action potentials faster than the parvocellular cells. And they're going to respond to that transient burst. You might think about, like, why would they want to respond to the transient? Well, that means they're going to be ready for a new stimulus. They're going to be ready for new stimulus quickly, meaning these are more about something's there, move on. It's not detecting new things, right? If you have a quick transient burst, that's going to be detecting something new that comes into your environment and getting ready for the next thing that's coming along. Whereas the parvocellular cells, those P-type cells, they make up about 90% of the ganglion cell population. They have a sustained discharge for as long as stimulus is there. So that's basically like saying, hey, yeah, something is always still there, right? So you're seeing an object, it's in your field of view, and it's still there, right? You see the table in front of you. The table is not going anywhere. It's sitting there. It's staying there, and that's going to activate these P-type cells. Now, if something slides across the table and something transiently goes through your field of view, right, that's going to activate these magnocellular cells initially, right? And then they'll shut down and be ready for something new to respond to. So two major types of cells, important to kind of keep track of those two things. Another thing about the retina, we're also going to have color ponate ganglion cells. So just like we have on-center, off-surround, we can also have a red on-center and a green off-surround, okay? Meaning that the red on-center, we put a red light on that middle, boom, we get a huge stimulus, right? We go red in the middle and red in the surround, okay? This is going to cause, that center part's going to cause the go up just like you did before. The red on the outside will probably cause a little bit of inhibition. Maybe not a ton of inhibition, but at least some. Because you can see the action potential slow down a little bit. But then we get into this red-green aspect here. And we put the red in the middle. That's giving us our excitation, a lot of excitation. But now we see that our action potential is reduced even more in frequency. Well, that tells me that I'm getting a lot of inhibition coming out of that green light. So you're going to have red, green, and blue, yellow kind of receptive fields, things that are going to offer contrast to different colors, and again, help let us see colors, let us see color edges, let us see some of the details, differences as colors are changing from the input into the retina. So one kind of last thing here, and we think about the retinal output. Again, you have simultaneous input from both eyes, right? We'll talk more about this in a little bit with the next unit, but we're getting the input coming from both eyes. They're going to get compared in a cortex, and that's going to allow us to do things like depth and find the distance of an object, right? Knowing how quickly it gets to one eye versus the other and the timing of the movements and things like that, that'll allow us to tell where that thing is in space based on how we see it at a retina. Again, you're also going to have information on light and dark. Those are off-center, on-surround cells. Okay, on-center and off-center ganglion cells. They're going to offer different outputs, and you're also going to have different receptive fields and different properties based on which type of ganglion cell you've got. Okay, some will have larger receptive fields responding to transient motion. Some will have small receptive fields that kind of keep track of things that are in your field of view all the time. So there's the basic introduction into how retinal processing is going to start. Okay? And then we'll get a little bit into what happens in the brain as things pass further on into those first couple to their first stop in cortex in our next unit.